Okay, good afternoon, students. Welcome to Biology Breeze. Yesterday, we have learnt about the process of fertilization in flowering plants, that is in angiosperms, that we recall as double fertilization and triple fusion, isn't it? And other subsequent changes that occurred. Now let us go into another important aspect, that is post fertilization changes. Okay, so what are the post-fertilization changes called? Now, all the events that occur in a flower after fertilization, all the events that occur in a flower after fertilization are known as post-fertilization changes. And what does they include? They include number one, the development of embryo because the zygote has been formed inside, so it develops into embryo. The development of endosperm, the formation of fruit, and the formation of seed. That is the maturation of ovules into seeds and maturation of ovary into fruit. Okay, so let us see the first aspect, namely the development of endosperm. Now, what is endosperm and how it is developed? So, since we recall yesterday that the second male gamete, when it was fused with the primary endosperm, that is from fuses with the polar nuclei, isn't it? So it forms a primary endosperm nucleus, which is commonly called pen. Now, what happens to this pen? The pen undergoes repeatedly what we call divisions, isn't it? Free nuclear divisions, undergoes successive nuclear divisions and give rise to free nuclei. And this is actually called as a, what is it called? It is called as a free nuclear endosperm. Free nuclear endosperm. Okay. And later on, the cell wall will be formed inside, and therefore the endosperm becomes cellular, isn't it? Now, I think you have already seen, or you have already have taken care about your coconut water, isn't it? The coconut water that you drink is free nuclear endosperm, isn't it? And the white color, the kernel that you see is actually the cellular endosperm. Okay, so this white, that is the white kernel, is a cellular endosperm, and the coconut water is called as a free nuclear endosperm. That's free nuclear endosperm. Okay, now let us see another aspect of this. Now, the endosperm may be completely consumed by the developing embryo, isn't it? Or sometimes it may be remain in that state. Okay, and especially in some plants like pea, groundnut, and beans, that endosperm may be completely consumed. Okay, endosperm may either be completely consumed by these plants, or okay, and similarly in some other plants like castor and coconut, it may be persist in the mature seeds and it can be utilized during the seed maturation. Okay, so that is why the preceding, that is the endosperm development, is a very important aspect as compared to the embryo development, isn't it? Because embryo is the baby of the plant, so the baby needs nourishment and food from food to grow. So from where does it gets it? So it can be able to get only from the endosperm. So therefore embryo development will be okay preceded by the endosperm development. So therefore endosperm development precedes embryo development. That is why it's a very important aspect of the what we call angiospermic plants. Let us go to second aspect, namely what is called embryo. Okay, now how this embryo is formed? We know the embryo is formed by this. Let us go to diagram 2.13. Okay, now look at here. We know that embryo is usually formed in the micropolar region. Why? Because that is where the egg apparatus is there, and the first male gamete combines with the egg to form the zygote. Now look at the zygote. That is 2.13, the stages. Okay, now look at the A. In A, we have seen how the process, that is a completely the process of fertilization is over. Now let us see the subsequent stages here. Now the zygote, which is usually okay, the nucleus of the zygote combines and forms one nuclei. It divides into two cells, the basal cell and the upper cell. Okay, now look at that. Slowly the cells are getting reorganized, and then it gives rise to, first of all, a proembryo. Okay, the two cell stage is called proembryo, followed by globular embryo. No, globular embryo because it is usually oval in shape or maybe round in shape, hence it is called as a globular embryo. And then we have got heart shape embryo. Okay, and then we have a mature embryo, and you can see in these stages In all these stages, you can see the presence of two cotyledons, isn't it? So that means these are the stages that will be mentioning in or will be seen in the case of a dicot embryo. Okay, so like that, slowly and slowly, the embryo is formed inside. Okay, so embryo, when you look at the embryo, okay, the development stages of embryo, which we called as a embryogeny. So what is embryogeny? The what do you call some the stages of where the embryo develops is known as a embryogeny. Okay, that is both in the case of dicots as well as in the monocots. Okay, now a typical dicot embryo. Now look at this figure 2.14. A typical dicot embryo has certain parts. Okay, so first of all, what does it has? It has got an embryonal axis and has got two cotyledons. You can see here. Okay, so the two cotyledons that we can see. Okay, here we have this diagram you can see. So you can see the presence of the two cotyledons here, and also we have the embryonal axis. Okay, so embryonal axis and the two cotyledons are the two important what do you call aspects of dicot embryo. Okay, the portion of the embryonal axis above the level of attachment of cotyledons, we call this as epicotyl, right? So what is a epicotyl? Actually, epicotyl is nothing but that will be represented by plumule. So you can see here that's a plumule. The plumule level later develops into a shoot system, right? The leaf primordia and other parts will develop from the plumule. And the portion of the embryonal axis just below the attachment of the cotyledons is known as a what you call a hypocotyl. What is it called? Hypocotyl, and that usually terminates into a radicle, and the radicle usually gives rise to the root system of the plant, where you can see the root is usually covered by a root cap which protects the tip of the root, as it has got a meristematic cells which will be giving rise to root. Okay, so these are the most important parts of a dicot embryo. So dicot embryo consists of an embryonal axis and the two cotyledons. Okay, then we have got the epicotyl, that is will be terminating into plumule, the hypocotyl that will be terminating into radicle, which will be giving rise to new plant. Okay, now what about monocot embryo? Let us see. That is 2.14. You can see the diagram 2.14 in B. There is monocot embryo. Now, as the monocot embryo, monocot, that is the term itself, help us to understand that the monocot embryo has got only one cotyledon. Okay, it is one cotyledon, has got one cotyledon, and usually see in members of grasses the cotyledon is not in the center, it is present towards the side. Yeah, it is situated towards one side of the embryonal axis, and that is called scutellum, right? We can see here in the diagram it is called scutellum because it is situated to the one side. Okay, now this embryonal axis has radicle on its lower side. Okay, you can see the radicle now, but the radicle is not what you call as exposed, it is covered by undifferentiated sheath. Okay, and what is this undifferentiated sheath called? This is called coleorhiza. Coleorhiza, right? So rhizoids means roots. So therefore, the coleorhiza is that part of the sheath that will be collectively, or that will be protecting the radicle from damage. So therefore, it is called coleorhiza. Okay, the upper end of the embryonal axis, okay, or the shoot apex is again covered by what you call by a few leaf primordia. Okay, and what is it called? It is called coleoptile. No, you can see. So in the case of monocot embryo, what you have to remember is, okay, that is it has got a single cotyledon, okay, and it is present towards one side, and it is called scutellum, right? The radicle is covered by an undifferentiated sheath called coleorhiza, and the plumule is covered by what you call another undifferentiated, or maybe is covering, okay, and that is called as a leaf primordia, and it is called as a coleoptile. Okay, so that is a very very important part of the monocot embryo. So dicot embryo and monocot embryo. So monocot embryo usually they will be asking in the examination. Okay, so that's a very very important thing that you have to note down, right? Now coming to the next part, namely called as a seed. Okay, so what is technically speaking the seed? Okay, what is actually seed? A seed is nothing but a fertilized ovule, right? A fertilized ovule is called as a seed, isn't it? Now, where is the seed formed? So naturally it is in the fruit, isn't it? Okay, it is formed inside the fruit, right? And the usually the seed has got three parts. Okay, what are those parts of the seeds? So we have got first of all the seed coats, right? One. Secondly, we have got the cotyledon, which may be one or two, and also we have got an embryonal axis or embryo axis. Okay, so remember the integuments of the ovule, isn't it? So since the seed is formed from the ovule, so what happens to the integuments? The integuments usually will become hard and they will be in protective in function, and they form usually the seed coats. So the seed coats are nothing but the ovules, what you call integuments. Remember we had studied about the structure of ovule, which has got an outer integument and inner integument. So those integuments will be combined to form what they call, they form the seed coats. Okay, and what about the micropyle? The micropyle will remain as a small pore. Okay, the micropyle will remain as a small pore. Okay, a small pore in the seed coat, which will facilitating the entry of oxygen and water into it during the germination. Okay, seed has to germinate, right? So this micropyle will remain as a small hole to which the actually the water and oxygen can enter. Okay, so that is a very important. As the seed matures, its water content will be reduced by 10 to 15%. Okay, and by mass, and the seed will usually become relatively dry. Okay, now usually what happens, some seeds, okay, they will be entering into a state of inactivity, and that is called dormancy. What is it called? Dormancy. What is dormancy? The inactive state of the seed is called dormancy, isn't it? And this dormancy usually it happens during unfavorable climatic conditions, but on the return of the favorable climatic conditions, the seeds become active and they start to germinate. Okay, seeds can also be, in the sense, it can be also be albuminous and non-albuminous, okay, which is called as a endospermic or non-endospermic. Now, what are albuminous seeds or endospermic seeds? Albuminous seeds or endospermic seeds are those seeds, okay, which retain a part of endosperm, okay, as it not used up during the embryo development. What is it? It is not consumed. No, actually they will albuminous seeds retain a part of endosperm and it is not used up during embryo development. Like example, wheat, maize, barley, castor, sunflower, etc. But in the case of non-albuminous seeds, there is residual endosperm, right? In the case of non-albuminous seeds, there is residual endosperm and it is completely consumed during the embryo development. And examples like pea, like groundnut, etc. Okay, and also you can see in some of the what you call seeds, right? In some seeds, like for example, in the case of pepper, now what happens? The residual, there will be a residual persistent nucellus. What is that? In some seeds, like in black pepper and also in beet, there is the remnants of nucellus persists. Okay, the remnants of nucellus. What is nucellus? The tissue, right? Inside the what do you call a megasporangium, the ovule. We have already learned about it. So therefore, you will find them. So that will be remaining. They will be remaining there. They will be persistent, and therefore it is called as a what do you call perisperm. So the perisperm is a very important activity of that. Okay, but you can also find, okay, in some other seeds, in the sense the seeds may be completely utilize their endosperm, which we call them as a endospermic seeds, and some of them may not be able to use them, called as a what you call a non-endospermic seeds. So that is of today's topic, children. Okay, hope you have understood all these essential aspects and the very important topics of the flowering plants. Okay, so stay at home, stay safe, okay, and also stay healthy. Thank you very much.